Hey, good morning. It's great to see you all. Great to follow Nathan's talk on total water monitoring. I love that term. It's exactly what we need to be doing. And this talk really also connects to Benit's talk a little bit earlier. So anyway, this is great. I want to share an idea that we've been working on, and hopefully you all can give some feedback and help make this better. Certainly welcome that. This work is, I'm just the spokesperson here. The more important people are my PhD student, Cole, and my undergraduate, Connor, has been working on this, as well as Andres Patrignani, who's here with us today, really the brain power on this project to develop Cropland Observatory nodes. If you're creative with your acronyms, you can pull CRONOS out of that, which is what we did. And it's a little bit, I don't know, surprising or ironic. One of the gaps we have in understanding soil water conditions is in our croplands, because it's quite difficult to put a long-term soil moisture monitoring station in the middle of a crop field. And if that station is, on the side of the field, that's not the same thing. So there's a lot of reasons, why farmers or researchers might be interested in having this information for forecasting crop yields, improving their management decisions, helping to evaluate and improve our models and our remote sensing algorithms. But most of our networks are not able to make soil moisture observations in the crop, which is what we need. And so just an example of why that might be a bigger deal than you might think it is. This is some of Andres's earlier work here. This map is a calibrated model that is showing modeled soil moisture conditions across one SMAP grid cell, one of these 36-kilometer grid cells we've been talking about. And this is in the fall of 2011. And I'm not showing land cover here. I'm just showing the soil moisture, but the imprint that you see, the red versus the blue, the red is dry, the blue is wet. That's the pasture and rangeland versus the cropland. So we're coming out of the summer, and because of some particular sequence of weather events, the winter wheat cropland, which is fallow during the summer, had been able to recharge in its soil moisture conditions, and soil moisture conditions were quite good across the winter wheat cropland in that region. But in the pasture rangeland, where we have warm season vegetation that was actively using water all summer, that soil was almost totally desiccated. And this very wet soil and this very dry soil may be separated by 1 meter. It's a human imprint onto the soil water conditions that we're not able to observe currently with any of our monitoring networks, nor with any of our current remote sensing tools. Although we're working towards remote sensing tools that might be applicable at this scale. So it's a big challenge that we're trying to address. And so we want to figure out how we can make a system that can easily be placed out into a crop field after planting and easily removed before harvest. And so that's what the motivation is for these CRONOS stations. This work funded by USDA. And similar to what Benita was talking about, we want to monitor the soil water conditions. In our case, we're using cosmic ray neutron sensing. I hope you're familiar with that. If you're not, find me afterwards. I'll explain it. I'm not going to take the time to explain it now, except to say that it's non-contact. There's nothing in the soil. Nothing's buried, because again, we've got to be able to take this thing in and out. So there can't be anything buried in the soil. So that's how we do this, the soil water content. Also, the beauty of that cosmic ray neutron sensing, you maybe mostly know, it's sensing a radius of approximately 200 meters in every direction. So it's sort of a field scale measurement, which is very nice. We use a digital camera, just looking downward at the crop to take those digital images, and we process them with an algorithm that Andres developed, the Canopeo algorithm that measures the green canopy cover of the crop as the crop grows and develops. And then we're using all-in-one weather station up on the top, as you also saw in Vinit's slides. All right, and so I just want to say about, a little data about how we evaluated these stations. These are what I'm calling the first generation stations that I'm speaking about here. We're running second generation stations now. We're trying to develop some third generation stations, trying to make them better. And so the first thing with the cosmic ray neutron sensing, you need to calibrate it. As with many of our methods, you need to calibrate it, and then you need to validate it in the field. And so that involves Cole going out and taking a lot of soil samples. And it's, working pretty well. We have here on the x-axis, the soil moisture from the soil sampling. The y-axis is what we're getting from our CRONOS station. And this is soil sampling the field around the station where the station is located. So we have a mean absolute difference of 0.025. That's pretty good in my experience. And here are the time series from these three sites. And so what you're seeing, the blue dots, those are the soil moisture estimates from the cosmic ray neutron sensing, the CRONOS station. You see some black diamonds with error bars. Those are the actual soil sampling, the field around the station. And then there's a dashed horizontal line that indicates the permanent wilting point, which we measured for that soil, for that field. The last thing I want to point out, there's a black solid line. That's the soil moisture value from the nearest Oklahoma mesonet station, just for the sake of comparison to show how these things might differ. And we saw at two sites what we expected to see. There's a large difference in the soil moisture that you might observe if you're looking at the nearest mesonet station versus what's happening in the field. And we expected that. What surprised me was this bottom one. If you look at this site, Bremen, down here, they actually agree quite well. So that's the one that's surprising to me, because normally we find like these other two sites. Now, another interesting thing we observed with the CRONOS stations, again, if you look at the blue dots, here at this Chattanooga site, you can see the soil moisture conditions as that wheat really begins to go through stem elongation and starts to advance in maturity in late March and April. The soil moisture conditions are drawn down until they actually drop below the permanent wilting point. And so that's telling us from the soil moisture, hey, this crop is in trouble. And from the soil moisture alone, we would say, yeah, this crop has probably experienced terminal drought stress. And then at Bremen, which is the northernmost site, we see that during the winter and the very first part of the spring, very little soil water use there at that site. Okay, so it's suggesting that the crop is not that active or not that well developed in terms of its growth. If we go into the green canopy cover, so these are the type of images that you get downward facing from that CRONOS station. So you receive that image and then you process it with the Canopeo algorithm, and it for every pixel determines if that pixel is green vegetation or not. And then we get a green canopy cover. And so for this particular image, it was 72.8% green canopy cover. And this was from the Chickasha site. And so you can do that multiple times a day, all throughout the season, and track the progress of that crop. And so those data look like this. So here we have on the y-axis the green canopy cover, and the x-axis is time. And you can see something interesting at Chickasha, this site. The crop was actually quite far along in green canopy cover, but then we got some pretty harsh winter conditions in December, And there was quite a bit of winter kill that knocked back the size of the canopy of that young wheat crop there. So that was interesting. You can see here at this Chattanooga site, it was sort of struggling all along. Southwest Oklahoma, very drought-prone region of the world, and this crop was struggling all along. And you can see starting in early April, the green canopy cover begins to decline. That should not be happening. That crop is not maturing yet at that point. And so that's that drought stress, and it never recovered from that drought stress. So again, we see a really good correspondence between what we saw on the soil moisture side and what we're seeing here. At Bremen, you can see what we saw earlier, that there was very little canopy development of that crop until well into March. That's why we saw that delayed soil water use that we observed in the prior slide. And also we observed at Bremen there, we could really see that, you know, our camera it's measuring about 6 square meters of the ground surface, but that 6 square meters we happen to be observing was not representative of the field. And so you see this discrepancy between the black diamonds is what we measured in the entire field, and our station didn't catch it. So I like the camera because it's got a larger field of view than the NDVI sensor, many times larger, but the camera is not large enough either, at least in this case, to get a representative sample. Okay, and then we can compare the CRONOS weather observations with the nearest Mesonet station. And what we expected, there's really not much correlation, at least for 2 out of 3 sites, between the soil moisture conditions we have in the cropland and what we're observing with our long-term monitoring network. For atmospheric variables, things like temperature and pressure, those type of variables, excellent agreement. As a matter of fact, I would say we probably don't need to measure those things in the cropland. They don't vary enough to justify that measurement. It's probably unnecessary. Other things like precipitation, you probably do want that local measurement. We observed some slight differences. These our all-in-one weather stations were given a slightly lower solar radiation readings than what we were getting from the Mesonet sites, and slightly higher wind speeds. And so that's one of the things we learned in this process. So next steps. So those first generation stations, because we were using helium-3, which is very expensive, about the whole package cost us probably about $20,000 per site. Now in the second generation, we're using lithium foil sensors for the neutron sensors. Those are lower cost, and we've got a Raspberry Pi. We couldn't find any good camera system on the market that we were satisfied with, so we built our own. Field testing, those are ongoing. Those stations we can create for about under $10,000 each. And what I would like to see is to get us to under $5,000 per station. We're experimenting with some folks from the physics department on a really low cost neutron detector, boron lined sensor, and then just sort of switching entirely, not having a data logger, just using the Raspberry Pi and an Arduino together as the data logging system, and also data processing. We can do a lot of onboard data processing. We're probably going to trim down the meteorological observations, and we're trying some creative approaches to packaging. One of them you can see there in the picture. So with that, that's ongoing. And so just again, acknowledge our USDA support, Rain-Fed Agriculture Innovation Network. We welcome your questions and feedback. And thank you very much.
Very interesting. I was thinking everything about the unsolved field. So what sort of time you are in the field?
Yeah, good question. So these these soils, I don't have that information here, but they tend to be silt loam at the surface and then pretty heavy clay loams at depth.
Do you think the constant flow or what's coming out?
So, yeah, great question. When we calibrate the neutron sensor, we do soil sampling around the field, and we just use the soil water data from those samples to calibrate the neutron sensor. And after that, it's sort of agnostic to the soil type. We don't have to get into any detailed characterization of the soil, except that point where I was showing the permanent wilting point. Yeah, that was from those same soil samples. So again, sampling at the field scale and then measuring the wilting point. We calculated the average for the field. Yes. Yeah. Yeah. Great questions.
You can just shout.
Thanks for that. You showed the whole correlation between your stations and the resident stations for soil moisture. And that's why I looked like the basic and the resonance, so you're very involved in doing growth straight. Generally, what's going on that?
Yeah, that's a great observation. If I can click back to it really quick, it has to do, yeah. So you see these vertical patterns on the x-axis where the mesonet is reading sort of constant, and so those at Oklahoma Mesonet we've been using heat dissipation sensors to monitor the soil matrix potential and convert that to an estimate of soil water content. But those sensors can lose sensitivity near saturation. So when we get close to saturation, they're just not very sensitive. And so you get some of that flat lining. It's a great question.
So are the soils actually saturated?
They're what we've observed is that they're not quite saturated. We just had a paper on this published. They tend to operate and hover around -7 kPa or 8, 9. They're kind of in that range. They're not quite saturated, but the fact that they stay in that range for so long has really shown us that we've got to stop thinking about -33 kPa as a field capacity. We need to throw that away. That's just not right.
I got time for one more question, and it's gonna be Ronnie because he's been racing. So
Thank you. Yeah, really great talk. Enjoyed it. Have you guys deployed have you co-located one of your CRONOS stations with the Oklahoma Mesonet to see if there's any sort of differences that might, the sampling versus the point measurement to explain some of the offsets between the field and the side field?
It's a good question Ronnie's asking about. We've co-located actually directly at an Oklahoma Mesonet site. We've done some of that in our preliminary analysis, but nothing that we've worked up to share. Generally, I would say if you put them exactly together, the correlations are significantly improved. Yeah.
all right, thank you, Tyson. We're gonna let you bother Tyson during the break if you have more questions. We got a couple things we need to get done. So thank you, Tyson. We have a 20-minute break because we're gonna have posters coming in to this room. So if you have posters, you can put them up in there. We have coffee in the back left over there. We're setting up snacks over here on the side. And I think that's everything. Be back at 10:30 local.